Welcome to The Explainer. Today, we're going to dive into what is honestly one of the most explosive and controversial medical mysteries of our time, the true origins of the Lyme disease epidemic. For decades now, there's been this intense ongoing debate. On one side, you have the established scientific consensus focusing on the natural evolution of tick-borne diseases. But on the other side, there are these deeply unsettling allegations that the U.S. government's Cold War bioweapons program actually played a role in engineering or at least accelerating the outbreak. In this explainer, we're going to impartially examine the source material from both sides of the aisle. We'll look really closely at the biological aspects, the alleged methods of weaponization, and most importantly, the hard mechanistic biology of how this disease actually spreads in the wild. All right, let's jump right in.
So let's get straight to the core question here. Did the military actually weaponize ticks? This whole idea really exploded into the public eye with Chris Newby's book, Bitten: The Secret History of Lyme Disease and Biological Weapons. Her investigation brought to light some pretty shocking allegations, specifically that the U.S. military conducted thousands of experiments exploring how to use ticks and tick-borne diseases as biological weapons, and that some of these agents might have accidentally escaped into the environment. The book relies heavily on personal files and interviews with Dr. Willy Burgdorfer, and if that name sounds familiar, it's because he's the very scientist credited with discovering the Lyme disease bacterium in the first place. Now you might be wondering, why on earth would the military want to use insects? Well, according to historical documents, arthropods like ticks inject pathogens directly into the human body. That means a standard-issue gas mask would offer absolutely zero protection to a soldier.
To really understand how claims like this could even exist, we have to rewind and look at the massive expansion of bioweapons during the Cold War. Back in 1945, under Operation Paperclip, the U.S. actively integrated former Nazi scientists into its ranks. One of them was Erich Traub, a virologist and a literal biological warfare expert. Then, by 1952, the Plum Island Animal Disease Center over in New York was officially transferred to the Army Chemical Corps to conduct biological warfare research. Fast forward to 1954 and you get Operation Big Itch. This was an actual military test where they dropped cluster bombs loaded with hundreds of thousands of uninfected fleas. They wanted to prove that insects could survive being dropped from an airplane and still successfully attach to hosts over a massive battalion-sized area. And then, in 1962, the government authorized Project 112, which was a sprawling, highly secretive bioweapons testing program that went on for over a decade.
Okay, now take a look at this staggering number, 282,800. According to declassified documents, that is the exact number of Lone Star ticks the U.S. military released across Virginia between 1966 and 1969. But here's the crazy part. They didn't just release them. They actually made these ticks radioactive with carbon-14. Why would they do that? Simply so researchers could walk around and track their migration patterns using Geiger counters. They were testing this biological vector release mechanism right along the Atlantic Flyway, which is the path migratory birds take up and down the coastline. Now, before these experiments, Lone Star ticks were incredibly rare above the Mason-Dixon line. But surprisingly, shortly after these open-air tests, established populations of these ticks started appearing up on Long Island. This essentially proved that laboratory arthropods could hitch a ride on natural wildlife and establish persistent populations miles and miles away.
But what exactly were they allegedly putting inside these ticks? Let's look at the specific allegations surrounding Willy Burgdorfer. According to the files uncovered in the book, Burgdorfer was experimenting with deliberate multi-pathogen tick infections. The goal was to turn these ticks into microbial mixing chambers, basically combining different bacteria and viruses to make the resulting illness much more virulent and way harder for doctors to diagnose. And the military's intent here wasn't necessarily to kill the enemy, but rather to create a slow-acting, completely incapacitating illness. The source material even points to testimony from a CIA operative who claimed infected ticks were literally airdropped on Cuban sugarcane workers in 1962 during Operation Mongoose. On top of all that, Burgdorfer's recently uncovered notes suggest he actually found a second pathogen in early Lyme patients. He called it the Swiss agent, a type of rickettsia bacteria. But the allegation is that he completely suppressed this co-infection research for over 40 years, purposely leaving it out of his landmark 1982 paper.
Now, if you're wondering how this ties into the actual Lyme disease outbreak, it all comes down to geography. And the geography is what really fuels this whole controversy. The Plum Island Animal Disease Center sits just 13 miles across the Long Island Sound from a town called Old Lyme, Connecticut. Old Lyme happens to be the exact location where the first 51 documented cases of what we now know as Lyme arthritis exploded out of nowhere in 1972. Plus, historical documents fully acknowledge that Plum Island frequently conducted outdoor experiments with ticks. The assumption at the time was that because it was an island, everything was perfectly contained. But as we know, biology always finds a way. Deer from Lyme regularly swam across the water to Plum Island, and local birds naturally flew back and forth feeding on insects. Biologically speaking, this created a direct, natural pathway for any potential laboratory pathogens to easily hitch a ride to mainland wild populations.
All right, but hold on a second. We have to pivot here, because the broader scientific and medical communities have some incredibly robust counter-evidence that directly challenges this entire bioweapon narrative. The most crucial sticking point is the timeline. It just clashes. The bioweapon allegations center heavily on government testing in the 1960s and 70s. But modern genomic surveillance tells a vastly different story. A really comprehensive study by researchers at Yale looked at the genomes of the Borrelia burgdorferi bacteria, the bacteria that causes Lyme, over a 30-year period. Through genetic sequencing, they calculated that this specific bacterium has existed right here in North America for at least 60,000 years. That is tens of thousands of years before human beings were even on the continent to build a laboratory, let alone engineer a pathogen.
So with that kind of genomic proof, you'd think the debate would be over, right? Yet, the mystery completely persists, and that is largely because of the man at the very center of it all. In 2013, near the end of his life, Willy Burgdorfer sat down for a really cryptic video interview where he confirmed his participation in bioweapons research. But it's what happened off camera that's famous. When the camera stopped rolling, he reportedly smiled at the interviewer and said, "I didn't tell you everything." On top of that, before he died, he left behind a note stating, "I wondered why somebody didn't do something." It's these incredibly ambiguous, late-in-life admissions that just keep throwing fuel on the fire and driving the public demand for declassified military records.
But let's step away from the cloak and dagger stuff and bring our focus firmly back to the hard biology. Dr. Sam Telford, who is a highly respected infectious disease researcher at Tufts University, firmly pushes back on this laboratory origin theory. He notes that the genetic data in the scientific literature simply does not support a point source origin. Basically, if this disease leaked from a single lab on Plum Island, the genetic data would show it radiating outward from that one specific point on a map. But it doesn't, not even close. Furthermore, Dr. Telford points out a very practical biological fact. The Lyme bacterium is actually a terrible candidate for a bioweapon. It reproduces incredibly slowly, and it is notoriously difficult to mass produce in large volumes compared to other, much more lethal agents.
And if 60,000 years wasn't enough to convince you of the deep biological history here, wrap your head around this number: 15 million. Back in 2014, scientists discovered a tick fossilized in a chunk of amber from the Dominican Republic that was 15 million years old. And when they analyzed it under a microscope, they found spirochete-like cells that showed clear evidence of a Borrelia infection. Moreover, researchers have actually gone into natural history museums and tested preserved specimens of white-footed mice from Cape Cod, dating all the way back to 1894. Guess what? They were already naturally infected with the Lyme bacterium. The hard biological evidence just overwhelmingly proves that this pathogen has been a natural part of our ecosystem for literal eons. It definitely wasn't just cooked up in a mid-century military lab.
So, if it wasn't a lab leak, what exactly is the mechanistic biology behind how this massive epidemic happened? Well, the scientific consensus points entirely to sweeping ecological changes. Let's walk through the steps of how this perfect storm formed. Step 1. In the 20th century, a lot of massive farms in the Northeast were abandoned, and those areas rapidly grew back into dense forests. Step 2. We humans actively eliminated natural predators like wolves and enacted strict hunting restrictions, which led to an absolute explosion in the unmanaged deer population. Step 3. We decided to build our sprawling suburbs right smack in the middle of these new forests. Ticks naturally expanded into these suburban landscapes, feeding on white-footed mice and robins, which happened to be excellent natural reservoirs for the Borrelia bacterium, and then they'd feed on the abundant deer. Basically, humans just walked right into a highly amplified, entirely natural biological life cycle. Throw in some warmer winters from climate change, which extends the tick's survival rate, and there you have it, the true biological culprit for the epidemic.
Now, even with all that solid scientific consensus regarding the ecological origins, the sheer volume of historical secrets from the Cold War has actually prompted real legislative action today. Representative Chris Smith of New Jersey authored an amendment to the 2026 National Defense Authorization Act that legally mandates a federal probe. This law directs the Government Accountability Office to conduct an exhaustive deep dive review of historical records from the Department of Defense, the NIH, and the USDA stretching from 1945 to 1972. Their explicit task? To definitively figure out if the government ever experimented with ticks as biological weapons, and if any of those weaponized ticks were released, whether by accident or by design. As Representative Smith has noted, regardless of what the biology says, the public simply deserves to know the absolute truth so we can finally turn the page on this chapter of history.
Which really leaves us with this final provocative thought. When the GAO finally opens up those government vaults, what are we going to find? Will the declassified documents completely rewrite our understanding of history, revealing a Cold War experiment that went horribly, tragically wrong? Or will they just confirm what the mechanistic biology and the ancient genomics have been loudly telling us all along, that nature, given enough ecological disruption and a 60,000-year-old bacterium, is perfectly capable of creating a devastating epidemic entirely on its own? It's an absolutely fascinating intersection of military history and evolutionary biology. Thank you so much for joining me on this explainer, and remember to always keep questioning the science around you.